Hi everyone, thank you for being here. Today I'm going to be presenting my research regarding Italian honeybee immunity. So to begin, honeybees are pollinators and they are in a mutualistic relationship with plants that results in cross-pollination. So in this relationship, the honeybees gain nectar and pollen from bees, from plants, and it is positively impacted on the bees so they can support their hive. And additionally, these honeybees produce honey that is beneficial to humans. And these bees produce about 160 million pounds of honey per year, valued at $300 million. So through these pollination services, they're incredibly essential to humans. So 90% of the world's food comes from just 100 crop species. Bees are responsible for pollinating 71 of these. So to picture this and put it in perspective, about 1/3 of our plate is attributed towards honeybee pollination. So this is an image showcasing what your plate would look like with bees and without. So without bees, we have no juices, no almond milks, jams, fruits, and etc. So these pollination services have economic contributions, estimated to be about $15 billion worth of crops annually just in the United States, with 1/3 of this being attributed towards almond cultivation. Now, this is an unique relationship because honeybees are one of the only pollinators of almonds. So every season when these almond trees start blooming, these honeybee hives shown here in these oyster boxes are transported towards California and they pollinate almond orchards to produce this product. So, there's a problem. In the winter of 2006, beekeepers reported that these worker bees were disappearing, for lack of a better term. And by the following year, about 36% of all colonies were lost. So this phenomenon is referred to as colony collapse disorder, attributing towards the sudden loss of worker bees, however finding very few dead bees. This is an image showcasing what a hive would look like before and after CCD. So, to put this on a global perspective, honeybee colonies, showcased here, are negatively accounted for in the continents of North America and Europe, in perspective to the rest of the world, with Asia being almost 300% of colonies. So this could be because of different factors, maybe agriculture or other reasons. So to focus on the United States colony loss, this graph here showcases years on our x-axis, and on our y-axis we have colony loss. So for the sake of this, we're going to pay attention towards the red bars representing annual loss of colonies, and we can see, unfortunately, an increased percentile loss in colonies for about almost a decade. So to represent this in the United States, we can see that the deeper red color represents highest colony loss. So in Texas, from 2024 to 2025, there was about a 61% loss of colonies. So, where are bees going and why is CCD occurring? Scientists would agree that this is a multifactorial and synergistic effect, with some of these factors being invasive species, parasites, viruses, habitat deterioration, and etc. For the sake of this project, we focused on pathogenic factors. So to understand these pathogenic factors, we have to understand Italian honeybee immunity. So for two different types of immunity, honeybees have an innate immune system, like many invertebrates. And this immune system provides a non-specific defense so when they impact a threat to their immune system, they attack and they are not able to remember these past invaders. On the other side, we have adaptive immunity, like many vertebrates like me and you have. This immune system provides defense, but it has the ability to remember past invaders to therefore influence how they respond to these impacts again. It also has the ability to learn from vaccines to help them fight these infections better. Also, innate immunity is understudied compared to adaptive immune systems. So, Putting all of this together, we formed our question. How quickly do these immune pathways in Italian honeybees respond when infected with a pathogenic threat? So to answer our question, we formed two categories of groups. We have our basal group, which is kind of our natural group. We fed these bees nectar or nothing at all. And on the other side, we have our activated immunity. This is our experimental groups. We fed these bees a lipopolysaccharide solution, or an LPS. So an LPS is a macromolecule found on the cell wall of gram-negative bacteria. We use this solution to stimulate pathogenic presence because it represents a non-self molecular signal. So this is how we're introducing the pathogen to our honeybees within our experiment. So with these groups, we would have an experimental group every 15 minutes, starting with 0 and going all the way to 240. We would have a new group every time. So to gather our bees, we would go out to our Langstroth-style hives, collect about 25 to 30 bees per group, and then we would feed them either the nectar or the LPS solution. And we would do this by administering the liquid through a micropipette tip. And she's going to ingest that. And we can make sure that she ingests the full volume by visualization. She's okay. And then after she is fed, she is incubated for the allocated time according to her group. And then we anesthetize our bee, and then we perform a dissection, and we isolate the thorax of our bee, so that middle section. And then with this thorax, we grind it up here with this pestle, and we get to the stages of our purified protein sample. So to do this, we would do processes of homogenization and centrifugation to get our purified protein sample shown here. And with this sample, we would run colorimetric assays. And to do this, we would utilize this plate shown here. We would add our sample, add our chemical reagent, and then use spectrophotometry to produce results that are attributed to color-changing enzyme interest to represent immune activity. So these colorimetric assays that we ran are prophenyl oxidase, or PPO, and lytic. Our PPO represents the melanin synthesis cascade. This cascade is essential to invertebrates, and essentially it produces melanin as an end product to encapsulate these pathogens to deter the pathogenic threat, with our lytic assay being the quantity of the lysozyme activity lysing bacterial cells to deter bacterial infections. So for our findings, we have our basal groups and our activated immunity on our x-axis, with our y-axis being prophenyl oxidase activity. So for our basal and our basal nectar groups, we see lower amounts of prophenyl oxidase activity. Essentially, these bees don't have the pathogenic presence, so they have lower activity. But the second we look at our 0-minute groups, this is an immediate upregulation of prophenyl oxidase activity, but it is also downregulated quickly within the following group. And we see this pattern followed throughout our experimental groups because this is essentially an energetically expensive response. Looking at our lytic activity, we have our same groups on our x-axis with our y being lytic. And looking at our first activated immunity groups, this is a delayed and decreased response, but it is upregulated following our 60 minutes, and we see this cyclical pattern followed throughout our experimental groups. So to look at these together, we see that our prophenyl oxidase activity is the primary means of response, and this PPO is activated immediately upregulation, but following the cyclical pattern, with lytic coming in secondarily as a kind of backup to fight these pathogens. So for our future directions, we're going to add a catalase assay, which quantifies the antioxidant capacity when reacting with a reactive oxygen species, or an ROS. This ROS is produced when the immune system is activated, but it can negatively impact their immune system. So when we add these three assays together and we look at them, we will have a holistic view of these honeybee immune systems to therefore understand how they respond to pathogens, so that maybe we can understand CCD better and try to help save these bees. So for my acknowledgements, I'd like to thank the USDA E3 and St. Edward's Biological and Natural Sciences community for supporting me in this research, and I'll take any questions. Hi, yes.
Hi, can you go to the lactic acid slide? So at 0, so like this is activity, so 0 should be 0. How do you have a bar in the negatives of activity for 0 minute?
So this response is very quickly. It responds, or this is activated fairly quickly. So when we run this assay, we run the assay over time, but we pull specific data points over the time scale. So when we put these together,
but how can you have less than 0 activity?
this
What are you measuring? What's the measure?
it's the rate of the lysozyme bacteria lysing the bacterial cells.
Right, but what's the actual measure? Cloudiness, like the live cell, like what's the data?
So this is the data of the color changing reading from the laser and the spectrophotometer. So like I said, we pull these 2 different points from our time point.
Is it possible that the base color of 0 can change, the color can change up or down from that? So it doesn't really mean less than 0 activity. It just means that what you're measuring, so it's kind of an artifact.
But then wouldn't the rest of them be artificially increased if that's not a baseline of 0? I know the question I'm asking is really tricky. I do understand that.
We're trying to take a technical thing and make it into biology.
Yeah.
That's what we're trying to
Because from a statistical point of view,
Yes, I apologize. It is a good question. I will have to defer to Dr. Stevenson to provide you a more concrete answer.
I'm so sorry.
No, you're totally good.
Are these data and the other ones you showed, this is multiple experiments? Like multiple individual bees?
So each group has like 25 to 30 bees. So we would get our bees, feed our bees, process our bees, and then have a new group. So these are all different.
And then did you do it like multiple days or like you did it once?
So
I'm wondering how repeatable this is.
all of our samples were collected around in the same summer. Yes.
But
they weren't done at the same day at the same time. You did multiple days at 0 minutes, multiple days at 15 minutes.
That cyclical pattern you see is repeatable.
Yes, this is what we were representing their immune system to be in what it would look like in one bee, but we did it with many bees. But we did not collect all of these bees on the same day. We would collect our bees, feed our bees, wait our time for one group in one day, and then process.
Yeah, I guess I'm just wondering if you went and collected bees today and did the same thing, would you see the same pattern? Or was it just, it happened that day? Yeah, it looks significant across the times, but if you do this repeated experiment over and over again, because your interpretation is that it's activated and it goes down. It's activated and it's going down. So that's true, you should see that tomorrow, yesterday, 3 weeks from now, right? So I guess that's kind of
Yes, that is what I would say we could predict, because based off of this pattern, we have enough data points up to 240 minutes. I would predict that the pattern would be the same. Hi.
I was a little curious. I saw on the slide when you were talking about the immunity and the adaptive immunity, there was one where it was like immunity that you could get from the mother, like breastfeeding. Do you know if for, I'm sorry, I don't know how this works for bees, but the queen bee is the one who's like everyone's mother, right?
Yes.
Do you know
Yeah, so this is an interesting question. It's a little bit out of the realms of this study.
Okay, sorry.
But no, the queen bee does help and is responsible for fertilizing all of these worker bee eggs, so potentially, but it's without the realms of this study.
Thank you so much, Hailey. I'm going to invite up Dr. Stephenson. We'll get some more yes, good. Go back to that photo. Dr. Stephenson is going to get to say a few things about getting to work with Hailey on this project, and I'll also present her with the coordinated stole that she'll wear at graduation to represent a lot of really incredible work in the honors program, and including but not limited to this study. So, Dr. Stephenson.
And I want to channel my inner Amy by answering your question, Chris, right?
Yeah, please. I want to know too.
Basically what happens is we have to choose particular time points, but there's so many bees that we have to look holistically at it, and we have to choose when the activity is the linear part of the curve. So what happens with some of the bees, right, is we're pipetting in with the multi-channel pipetter. There can occasionally be some air bubbles in there, and what it does is it creates an air bubble that interrupts the laser, so it looks like we have massive activity, and then it drops really quickly as soon as the bubble pops, and then we get the linear part of the curve. So there are a certain number of time points that look negative, and it draws everything down, and we kind of get those bars like that occasionally. And there's really, unfortunately, nothing you can do about that. Yeah, so we just have to choose the best part of the time point so we don't miss the activity, but we also don't want to get that, but we do occasionally. So that answers that.
Really technical, I'm so
sorry. So I don't know, I'm sure Hailey remembers, but a lot of you might not know, is that Hailey was actually in my Gen Bio class as a freshman. So Hailey was in my class, and I'm gonna embarrass her a little bit. She took the first exam, she did okay, right? She did okay.
That's generous.
But she was one of the only students in my Gen Bio class that came to my office and she was like, okay, how do I do better? What do I do? How can I earn a higher grade? She consistently came to office hours, she engaged throughout class, and she ended up in her particular group kind of leading every single discussion that we had whenever we did like, I think, pair share or like an in-class activity. So I asked Hailey to do research with me when she became an EBCC major, and so she joined my lab. And I know we kind of alluded to this, right, in the data, and I don't know if anyone heard, but Dr. Rowe behind me said, wait a second, so how many bees did you kill, right? This is the largest data set that I have ever produced in my career. It is massive. It has taken 6 students 2 years to complete all of these assays. And it's absolutely insane. We have gone through more pipette tips than you could possibly imagine. And through it all being this massive, massive data set, we have had Hailey who has been leading the charge, right? She has been the one that has said, hold on, we're gonna organize this. I wanna know what date we did this. I want this spreadsheet to do this. And she kind of literally organizes everything. And 2 of her fellow students are here and they're shaking their heads vigorously. And so Hailey has been absolutely instrumental. And I knew that she was going to be an amazing student when we had our very first research meeting, and she showed up to my door 10 minutes early, and she knocked, and she was like, are you ready for me? And I was like, we're not meeting until 2. And she looked at me and she said, on time is late. And I was like, okay, then this is gonna work out. So here we go, right? And then I do have one more small gift for you, actually. Over the past several years, you and your fellow compatriots have terrorized me, right, as your mentor. And so they decided to return the favor by working collectively with another member of our audience, I might point out, to elect me for the Kosmetsky Last Lecture Series winner, and they drummed up support to make me win it so that I would have more work to do by creating an additional lecture. And so Hailey was selected to be an oral presentation, in a competitive oral presentation at a national conference, and she was not able to go to my Kosmetsky lecture. And so I do have this right here for you. Just so you can remember, you can take that home, that's yours, right?
I don't think it'll fit on
just
my
be
fridge.
careful with your hands, don't break it, you know what I mean?
Thank you. I don't know what I would have done
I know,
without
right?
the science of models.
Can you take a picture?
Yeah.
Yes. Oh, we gotta have this.